Hello guys, my name is Suraj Katam and today we are discussing on genetic engineering. Genetic engineering is a process that uses laboratory based technologies to alter the DNA makeup of an organism. Genetic engineering also called genetic modification. This may involve in changing a single base pair or deleting a region of DNA or adding a new segment of DNA. The next one is where genetic engineering used for. Genetic engineering allows scientists to move desired genes from one plant or animal into another. Genes can also be moved from an animal to a plant or vice versa. Another name for this is genetically modified organisms or GMOs. There are some types of genetic engineering and that are DNA isolation, gene cloning, gene design, transformation, transformation, and fifth is plant breeding. And the examples: plants that might fight population, golden rice, faster growing trees, bigger or long lasting tomatoes, insecticide corn, non carrying onions, or cloning examples. Genetic engineering can be applied to any organism, like from virus to a sheep. For example, genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. Next one is genetic engineering may involve adding a gene from one species to an organism from a different species to produce a desired trait. Used in research and industry, genetic engineering has been applied to the production of cancer therapies, brewing yeasts, genetically modified plants, and livestock, and more. Genetic engineering has changed over the years from cloning for analysis and laboratory use to truly synthetic biology for understanding and new biomedical capabilities. The next one is genetic engineering is accomplished in three basic steps, and the steps are there: the isolation of DNA fragments from a donor organism. Second one is the insertion of an isolated donor DNA fragment into a vector genome, and third one is the growth of a recombinant vector in an appropriate host. The next one is a genetically modified organism. GMO is an animal, plant, or microbe whose DNA has been altered using genetic engineering technique. For thousands of years, humans have used breeding methods to modify organisms, like corn, cattle, and even dogs have been selectively bred over generations to have certain desired traits. There are some positive impacts, like some benefits of genetic engineering in agriculture are increased crop yields, reduced costs for food or drug production, reduced need for pesticides, enhanced nutrient composition and food quality, resistance to pests and diseases, greater food security, and medical benefits to have the world's growing population. Genetic engineering, also called as genetic manipulation, and the manipulation is the modification and manipulation of an organism's genes using technology. It is the set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is obtained by either isolating and copying the genetic material of interest using recombinant DNA methods or by artificially synthesizing the DNA. A construct is usually created and used to insert this DNA into the host organism. The first recombinant DNA molecule was made by Paul Berg in 1972 by combining DNA from the monkey virus, that is SV40, with the lambda virus, as well as inserting genes. The process can be used to remove or knock out genes. The new DNA can be inserted randomly or targeted to a specific part of the genome. Next one is an organism that is generated through genetic engineering is considered to be genetically modified. It means GM, and the resulting entity is a genetically modified organism, GMO. The first GMO was a bacterium generated by Herbert Boyer and Stanley Cohen in 1973. Rudolph, Rudolph created the first GM animal when he inserted foreign DNA into a mouse in 1974. The first company to focus on genetic engineering, Genentech, was founded in 1976 and started the production of human proteins. Genetically engineered human insulin was produced in 1978 and insulin producing bacteria were commercialized in 1982. Genetically modified food has been sold since 1994 with the release of the Flavor Saver tomato. The Flavor Saver was engineered to have a longer shelf life, but most current GM crops are modified to increase resistance to insects and herbicides. Also, the genetic engineering has been applied in numerous fields like research, medicine, industrial biotechnology, and agriculture. In research, GMOs are used to study gene function and expression through loss of function, gain of function, tracking and tracking and expression experiments. By knocking out genes responsible for certain conditions, it is possible to create animal model organisms of human diseases as well as producing hormones, vaccines, and other drugs. Genetic engineering has the potential to cure genetic diseases through gene therapy. The same techniques that are used to produce drugs can also have industrial applications such as producing enzymes for laundry detergent, cheeses, and other products. It means gene therapy technique that are used to produce also it produces drugs or some industrial applications such as enzymes for laundry detergent, cheese, and other products. Next one, the rise of commercialized genetically modified crops. So there is another definition for genetic engineering is the process of inserting new genetic information into existing cells in order to modify a specific organism for the purpose of changing its characteristics. Okay, here we have some steps, multi-step processes for creating a genome. Creating a GMO is a multi-step process. Genetic engineers must first choose what gene they wish to insert into the organism. This is driven by what the aim is for the resultant organism and is built on earlier research. Genetic screens can be carried out to determine potential genes and further tests then used to identify the best candidates. The development of microarrays, transcriptomics, and genome sequencing has made it much easier to find suitable gene. Also plays its part. The Roundup Ready gene was discovered after scientists noticed a bacterium thriving in the presence of the herbicide. Next step is inserting DNA into the host genome. There are a number of techniques used to insert genetic material into the host genome. Some bacteria can naturally take up foreign DNA. This ability can be induced in other bacteria via stress, which increases the cell membrane's permeability to DNA. Uptaken DNA can either integrate with the genome or exist as extrachromosomal DNA. DNA is generally inserted into animal cell using microinjection, where it can be injected through the cell's nuclear envelope directly into the nucleus or through the use of viral vectors. Plant genomes can be engineered by physical methods or by use of agrobacterium for the delivery of sequences hosted in tDNA by binary vectors. And now the last one is the applications of genetic engineering. It has applications in medicine, research, industry, and agriculture and can be used on a wide range of plants, animals, and microorganisms. First one is the bacteria. The first organism to be genetically modified can have plasmid DNA inserted containing new genes that code for medicines or enzymes that process food and other substrates. Thank you.